Well, hello AP Biology guys. I'm going to be demonstrating meiosis and how this works. Now remember, this is not mitosis, this is meiosis. So remember, mitosis is all about how cells divide each other and they go on and they just make exact copies of each other and clone each other. But in meiosis, there's more of a purpose of reproduction here. So instead of the cell just copying itself, these are specialized cells that are either going to end up making 4 egg cells or 4 sperm cells, okay? And so it all starts with G1 when the cell gets the signal to divide and to make either egg or sperm. And what will happen is the cell will prepare for DNA synthesis. So it's going to accumulate a lot of different bases, a lot of different nutrients and things to get ready to duplicate its DNA. Then it will enter the S phase, and that's when these single chromosomes, remember this is like a single chromosome, and so far we have 4 single chromosomes in this particular cell. But during the S phase, remember that the DNA gets replicated or duplicated, and they call it the S phase because it's called DNA synthesis. So this DNA will be copied, and we'll get an exact copy of DNA here, here, and here, all during this phase. And as you can see, now we have duplicated chromosomes where the left side is an exact copy of the right side. And remember, we can also call these sister chromatids as part of each duplicated chromosome, okay? Now, keep in mind that the human body cells normally have 46 of these chromosomes. And if we had 46 chromosomes in our model right now, we'd have chromosomes all over the place. So right now, we're just going to deal with 4 chromosomes. But we've got our chromosomes duplicated and we're ready to go to the next step, okay? So then we go to G2, and it's more preparation for cellular division. So more nutrients will be gathered and getting ready for division of the cell. And then it will enter meiosis. And so the first step of meiosis is prophase 1. Now remember, meiosis has a double set of divisions, okay? So we're going to go through 2 rounds of divisions here. And the first step of the first division is prophase 1. And during prophase 1, these duplicated chromosomes will condense and coil and become more visible underneath a microscope. So the DNA in each of these chromosomes will coil up and become nicely packed. But also, the chromosomes will pair together, and they'll form these homologous pairs. The word homologous literally means same-sized pairs, okay? Some other things happen. Not only do they pair up, but they can also be called tetrads. The term tetra means 4. In other words, in this pair of chromosomes here, we've got 4 copies of DNA, so we call these tetrads as well. Now, while these tetrads are paired up, another really interesting thing happens. You see, and this actually helps explain why siblings look so different from each other. And that is because before the egg and sperm are created, during prophase 1, there's an event that's called crossing over. And during crossing over, that's when there's going to be an exchange of segments of DNA, an exchange of genes between the adjacent sides of these chromosomes, okay? It happens right along the adjacent sides. So you see, now we actually have 4 kind of different shuffled copies of the DNA. Now before this, you remember you had half your chromosomes came from your mom and half came from your dad. But this is the point where the genes actually get shuffled up, and it's a good source of diversity. You'll see in meiosis that there are actually 3 big sources of genetic diversity, and this crossing over is also known as the first kind of place where the diversity comes from. One other thing, that is where they cross over, they kind of make a little X shape like that, and the symbol for X in Greek, I believe, is chi, and so they call this chiasmata as well when they cross over like that. But let's say some DNA has been exchanged. It can happen in all the different chromosomes, just along the adjacent sides within each tetrad. But then we're going to go on to the next step. And the next step is metaphase 1. And so during metaphase 1, the chromosome pairs will line up. These tetrads will line up. And remember, the spindle microtubules have already attached at the kinetochores of the chromosomes. So they're already pulling and tugging the chromosomes around. But during metaphase 1, they line up just like that. And when they line up, This is actually the second source of genetic diversity, because what can happen is they can actually line up on either side of the equator of the cell, okay? It's kind of a random alignment along each side, okay? So that's metaphase 1. We got everything lined up. During anaphase 1, the spindle microtubules will tug on these kinetochores, and they're going to pull these tetrads apart. So now these chromosomes can go to opposite sides of the cell, okay? Now think about it for a second. At this time, the total chromosome number has been reduced in half. So now we've gone from diploid to haploid cells, from 2N to N, okay? And then at telophase 1, we'll have cytokinesis, or a pinching of the membrane in the middle of the cells, and we'll end up with 2 new cells then. But remember, we're not done. We're going to go through a second round. So we'll go through prophase 2. During prophase 2, the chromosomes just remain coiled. There's not much changing going on. Then during metaphase 1, or metaphase 2, the chromosomes line up again, but you see now they're lined up differently. They're lined up so the centromeres of the chromosomes are right along the equators of the cell. So that when they do align, they can still align kind of randomly like this. They can be kind of on either side, more diversity happening. So that still counts as the second way that diversity can happen, is the random alignment during metaphase in meiosis. Then during anaphase 2, the chromosomes get pulled apart again. And this is the time when these sister chromatids get pulled apart and they officially become daughter chromosomes, like that. And so they get pulled apart. And so now we have telophase 2, and during telophase 2, there's more pinching of the membrane, more cytokinesis in each of these cells. And now we've got 4 cells. And notice how each of these 4 cells are genetically different, okay? So in the case of a male, these would be like 4 sperm. In the case of a female, they would be like 4 eggs. And notice how they have different combinations of genes, even though they still have the number of chromosomes they need. They have haploid numbers, so that when they go and recombine with the other opposite sex cell, they'll have a complete set when they're all done. One thing to just note, remember, if the chromosomes were not doubled in the first place, we'd end up with only half the DNA and things wouldn't work out. So that's why we need DNA synthesis to happen way back in the S phase like it did, okay? Oh yeah. Then there's the third way that we have genetic diversity happening, and that is that any one of these sperm in a male can combine with any one of the eggs in a female at random, and that is a third source of genetic variation, which goes on to make, you know, different brothers and sisters. And so the only way that you can have identical twins is if it comes from the exact same cell in some fashion, okay? So in summary, we have this end result after a double division of cells. We have 4 genetically different cells that all have a haploid number of chromosomes. Thanks a lot, guys.